So it's a pleasure to have here Professor Danilo De Masci from the Politecnico di Torino. We work together on different projects since a while, and he was on the way from Grenoble and come today to us to present his research on carbon nanotubes and also more a general overview about the institute in Politecnico di Torino.
So, thank you, Silvia, for the invitation. It's a great pleasure to be here and to have the opportunity to, well, from one side, to show you the activities we are doing in the use of carbon nanotubes or in general carbon-based materials for this technique that is named ElectroChemiLuminescence. I will start my talk introducing you the lab in which I'm working, which I'm responsible, so I will show you which is the environment in which we are working, and then I will pass to enter in details in the explanation of what is electrochemiluminescence. I'm not a chemist, so I will talk about the application, the use, again, of this technique for building sensors. So, if you remember, the title is for sensing. So, to use this technique for building mainly biosensors. And I will introduce all the ECL, the methods and the system that we realized and we are realizing, the test we did starting from the more classical way, so using metal electrode, and in this case we use mainly gold, also some testing with platinum, because this step is important to see after which can be the plus, the advantages we have using carbon in general. I will spend a few words in details talking about the system, the electronic system, the management of the experiment, and then I will pass to the part in which I will focus more, because this course is related to carbon nanotubes. So I hope to be able to show you which are the advantages, which are the possibilities we have in the use of carbon nanotubes for electrochemiluminescence, and also I will show you some preliminary experiments and tests we are doing for using other type of carbon-based materials. And then I will conclude my talk. Where I'm coming from? I'm coming from, as Silvia told you, from Politecnico di Torino, in particular from the lab named MINES, Micro and Nano Electronic Systems. This lab is new because it was born in 2010, but is a sort of evolution of of the same lab in terms of spaces, but we decided to change a little bit the name, because this lab was named a few years ago KeyLab, and it was a shared idea starting from 1999 between a consortium in the area, in the region, the Politecnico di Torino and the National Institute of Material Physics. So is up to now a shared laboratory between the Department of Electronics, from I'm part of, the Department of Material Science, now Applied Science, because we changed a little bit the names of the departments in Politecnico, but the competence is related to material science, and this institute named Istituto Mario Boella, I don't know if you know it, is an institute focused on wireless communications, so in telecommunication systems, but in particular wireless. And so MINES is the natural evolution of this lab, because in this lab we set up, and I will show you some details later, clean room and all the facilities around the clean room able to do what was in the minds of the two founders, so Professor Pierluigi Civera from electronics and Professor Fabrizio Pirri from material science department. They had the idea to set up a lab in which it was possible to do all the different steps from starting from the MEMS, from the device, up to the milling machine for producing the box, for producing what you need for using. So in the middle you have, starting from the device, you have electronic interfaces, the software, different level of software, the use of the software, and then the production of, we are not a company, we are not talking about products, but we are talking about prototypes, working prototypes to show that some of the ideas we have or other colleagues have can be useful, feasible, and they can work. So, from this side, now we are passing into the nano world, and so this is why we decided to change a little bit and to open also the idea of the lab to the new nano possibilities. So this is why was born the MINES, Micro and Nano Electronics System. The activities we are doing there, just some shots on it. We are talking about nano devices and systems for nano, of course micro, electronics and sensing. The design of microelectronic circuits for, as I told you, the interface, the management of the signals we receive from the sensors, for example, and also some basic electronic PCB boards, firmware, microcontrollers for having the possibility to create a complete prototype. And so, in this part, this is not a trivial, but is a very complex, long, expensive part to put all the things together and let them work. So do the system integration and also the packaging, because I'm sure that you know it very well that packaging, talking MEMS and now NEMS, is one of the most important part. Also talking about money, talking about the cost of the final product, because quite often it is more expensive, the package, than the device itself. And so you must put a lot of efforts for finding the most efficient, the less expensive package for managing your device. And we are also working on, as I told you, and today in particular we will talk about biosensing and the needed microfluidics. We are not, let's say, expert, we do not do research in microfluidics, but for us is a sort of tool, the microfluidic, that is needed for managing, controlling the liquid and the samples we are using for the biosensors. So, the lab has two sites, one in Torino, but I can say the main site in a small, it is in a small town close to Torino named Chivasso, and well, total of this is the area, and we have one clean room, small clean room, 80 meters square, where we arrive up to have a cleanness of 100, okay, 100 and 1000, which is the, well, the professor, the assistant professor who are participating, a PhD student, to the lab. And this is just a few shots about the clean room, managed by my colleagues from material department. They are the responsible of the clean room. And in this case, I show you, we have this class of 1000 where we have some, for example, we have an evaporator. After I will tell you a list of the possibilities we have. We have a 100 part here where we have a photolithography part, and on the back here we have a class 10,000 where we have some dirty works, dirty machines, the back side of the machines for avoiding the pollution to enter in the clean room, and these are some photos of the clean room. So, as I told you, the idea is, yes, I want to start from designing, and in this case we are the first involved, and in this case I ask to my colleagues responsible of the kingdom to build the devices, to build the MEMS we could have in mind for the production of our applications. I can tell you that the precision we can reach in our kingdom is not so high. I mean, we arrive up to a resolution of 5 microns. We are building for few applications also devices with 1-2 microns, but to be sure to have a good reproduction of the results, I can tell you that we can stay around 5 microns. That for MEMS is not too bad. You can do a lot of possibilities, a lot of applications, a lot of devices. So, a list of some instruments we have here. You have also the photo of the E-beam. So we have a standard lithography, but also the possibility to have E-beam lithography. We have a PECVD. We have benches for doing dry. Also we have a deep reactive ion etching, and also benches for doing the wet etching. We can do anodic and wire bonding, electroplating. We have also SEM microscope, in particular FE SEM microscope, other type general optical microscopes. This is the new arrival in the lab, a dual head microscope AFM plus STM, and all the tools, in this case I'm the one responsible of it, all the electronic hardware software for the design of electronic part and the related CAD tools. Okay, so this is just an overview of what we are doing there, and in particular now I'm also here for the cooperation we are having with Professor Schinke here in the development of courses in the field, in the focus on nanoelectronic devices. I don't show you in details the nanoelectronic activities we have because it's not the topic of this course. Just I want to tell you that in particular in this nano health and nano skill project, I'm giving the expertise we have in the production of nanogaps, so in the production of electrodes with a nanometric distance between the two electrodes, and so with these very, very, very close electrodes, you can produce both devices for molecular electronics, because you put in the middle organic molecules for building new styles, new ideas of devices for electronics. These molecules have a length of maximum 2-3 nanometers, so this is why you need so close electrodes. From the other side, the same solution, the same electrodes can be interesting in production of sensors, because you can put in the middle molecules. For example, we are working in particular in protein sensors, and I have some images, some results in which we have one protein inserted in the middle of the electrodes. And so you can build what are named single molecule sensors, very interesting for studying not a population of molecules, not a population of million or thousands of molecules, but just the single or very few molecules. So this is what we are working together in this part. From the other part, and this is why I'm here, we have in this lab that I recently showed you these two large projects, the nanogap, molecular electronics, sensing, single molecule sensor, and also from the other side, this project that it started during my PhD, so very long time ago, when the horses were around the towns, okay, no cars around, but it's I discovered, I read in, because I did my PhD in the Children's Hospital of Philadelphia, part of my PhD, and well, looking around papers, I found this technique, the ElectroChemiLuminescence, that was really interesting for me, because my boss, Professor Civera, sent me to Philadelphia, to the Children's Hospital. I'm an electrical engineer, so he sent me there to study the possibilities for microelectronics guys to give solutions, to give ideas to the, at that time, where I'm talking about 1991, 1992, the biochemical, the PCR, so the replication of DNA, all these activities were almost at the start. And so the idea to build micro devices for entering in this field was a really new topic. And so I went there for studying the possibilities we had. And this is why, in the meantime, I also found in literature talking about the professors there, this technique. This technique, the ElectroChemiLuminescence, I will use more ECL now. It is interesting from my point of view of talking about me that I want to build the application. It's interesting because it's a sort of competitor, we can call it like this, of fluorescence. Fluorescence means I have my molecules on the surface, bonded, linked in some way with biochemistry, so reactions here. I have my labels here, and using a laser source, I stimulate my molecules, cyanine or other fluorescent dyes, and I illuminate them, usually, for example, for argon lasers or blue light almost. And when they are stimulated, they emit a light with a different wavelength, of course. I have a difference in energy of emission, and the emission can be in the red, in the yellow, okay? So, for example, I induce my emission using a blue laser, and I have back from my molecule, for example, a red light.
But you never change the wavelengths of the laser. You just change the dye, and it will change the wavelengths.
You are perfect right. Well, of course, there are several possibilities of lasers. Okay, I'm telling you the most used,
okay.
But you can have different light of lasers. But if I build one instrument, I do not change the laser. Okay, I use this laser. In fact, and your question is interesting because talking about DNA, you know, DNA have 4 bases. And in fact, the biochemists, they are good people, they built up 4 different dyes with 4 different colors. So they are able to also detect, depending on the color, which is the base that you label. There are also very complex, I don't know if you are expert or not, but the concept is based from the fact that if I have green light, means that I have a thymine. If I have a yellow light, means that I have a guanine. So they do what is named sequencing, they do different reactions. So exploiting the fact that depending on how you label, with the label you use, and you use the label, the red, it sticks only on the cyanine, of course. The yellow sticks and bond only with the thymine. Okay? So, in this case, I have a possibility also to reading the colors I have in front of me, understanding how is, for the, you know that recently, now past a few years, they discover all the sequence of human DNA. Okay? And they use these techniques, these optical techniques. Fluorescence. So, I have emission, but you can imagine, I have a laser, so a very strong light. The power of a laser it's high, of course, not too much, I don't want to burn my DNA, but it's the emission of a laser have a very strong light. The emission of the molecules, and also, always, I want to reach the minimum detection limit with very few molecules, because I don't want to use too many reagents and so on. So, what happens? It happens that the emission of what I have on the surface is lower than the power, in terms of light, of the stimulus of the laser. So, what I have to do here is to put, before having my light detector, photomultiplier, photodiode, or what is, filters, optical filters, to filter out the light of the laser. And so to have only reaching the surface of my light detector, only the emission of the molecules I want to detect. The ECL is interesting because the emission, and we are talking always of light emission, luminescence, it is induced by an electrical field. So, I put an electrode on the bottom, I attach my probes, I attach my molecules on the surface of the electrode, and I give to the electron, and we will see in details what I must do for inducing the emission, but now I want to start to give you the concept. I give the correct voltage to my electrode, and with this specific voltage, my molecules here emit light. This is great, because I have the two things, stimulus and emission, in two totally different domains of physics. So, I do not have at all interaction or problems of having a mix of signals. All the light that I have in my chamber, these experiments, of course, you do them in dark boxes, in closed boxes, in dark, and all the light I have is the one arriving from my molecules, from my labels. So you can, I hope, imagine how much can be interest this technique, because, and it is proved, you can reach lower detection limits. You can go down in detection limits because you are not blinded by laser, and it costs very, very much less, because no laser, no filters, not great infrastructure. I just need one electrode. Well, for the emission, and after what we will see together, a complete electrochemical cell for managing the experiment. But the cost of it is very much less of the one of fluorescence. So, this is why we decided to start to work and to exploit this technique. Let's go a little bit in details. So, but what happens to my level, to the molecule that will emit the light that I use to detect if my molecules are on the surface of my electrode? There are several molecules. The most famous, most used, and the one we decided to use is the ruthenium 3-bipyridyl. 2 plus 3 and 3 plus, but you start from this molecule. Okay, we are talking, I already mentioned, of an electrochemical cell. So we have reduction or oxidation, so redox reactions inside this cell. What does it mean? It means that on the surface of the electrode there is an exchange of electrons, between the electrode itself and the molecule that I'm detecting. In this case, the ruthenium trisbipyridine loses an electron, so it oxidizes on the surface of my electrode, and there is a cyclic reaction from which, after that it oxidizes, it passes to another type of this group, and then after this, it is not a stable situation of the molecule, so it comes back to the starting point, emitting a photon. Here, in this example, in this application it is, because it is the one we are using, there is also the 3-propylamine that is a co-reagent that helps, increases the efficiency of the reaction. So, what I'm interested in is the reaction of ruthenium trisbipyridine. The TPA, it's something that increases the efficiency, and so very useful, because I can tell you, without the TPA, of course you lose emission power, so you cannot reach detection limits, interesting detection limits. But here, electron, because I have the electronics, the exchange of electrons between electrons and the molecules, the chemical energy, the chemical reaction inside the system, and then the luminescence, the emission of the light. Good? So this is the concept. And the emission of the light, it is now talking about ruthenium, about it's an orange light, 620, 622 nanometers in wavelength. So I need to set up, the setup of my system, it is an electrochemical cell. And the electrochemical cell you use in these cases, it's a free electrode. You have your silver, well, if you are expert or you know electrochemical cells, the standard for reference, this is the reference electrode of my electrochemical cell, it is usually a silver chloride electrode. But we want to integrate, we want to create microsystem, microelectrodes for this application. The silver chloride electrode, it is very large, complex, and expensive. So we decided to use silver for our reference electrode, because from one side is not the standard, you do not have emission at maybe the standard voltages you expect, but I will show you that it's not so important for us to have emission in a standard way. But using silver, you can imagine, we can integrate the electrode very easily. Now after, this is what is named a counter electrode you need. The counter electrode, we made it in gold. And then you have this one, the third one, that is the working electrode, that is the electrode on which you have the reaction I show you here. So these reactions happen on the surface of the working electrode. They happen also on the other, on the counter electrode, but with a less efficiency because of the system. So, but we are interested in detecting what happens on the surface of the working electrode. On the working electrode, we can use different materials, and this is the key that I told you when I started. We started using metal, and I will show you examples using metal electrodes, and then, this is the final solution I want to show you, we passed at the end to use also carbon nanotubes or carbon-based in general electrodes as working electrodes. The rest of the systems remain almost the same. or we chose to leave it as it is because of doing comparison, with changing only the working, all the system, the rest of the system stays the same, remains the same, and so I have the possibilities also of comparing the different solutions. So, talking about the electrochemical cell, I need a potential state, so the electronics for managing my electrochemical cells. And from the solution, we are interested in detecting the emission light, the light emitted by the working electrode. And here, now we are using a photomultiplier for detecting the emitted light. All of it is managed by a computer, a workstation. Okay? Please.
You detect only the intensity of the emitted light or also the wavelength?
No, only the intensity. Also because in this case, your question is right because it's related to the previous question. Up to now, we are working only with this label. So we have only one wavelength. You're right. And also in ElectroChemiLuminescence, the biochemists develop different labels emitting different lights. In that case, you need to also have a detection in terms of wavelength. But now, up to now, we did not start this, and we are focused on this molecule, this group of ruthenium.
But in fact, the wavelengths, the emission wavelength is defined by the molecules.
Yes.
So
this molecule emits...
You just have to take the intensity because you already know the wavelength.
Absolutely.
Okay.
Absolutely.
But in some years, you will be able to detect also the wavelength.
In case, but in that case, the solution can be to insert filters.
Okay.
Optical filters, because it's a unique point. So we come back to the need of optical filters. You spend more, but it is. And the wavelength of emission is strictly related to the molecule you are using. So we are using that molecule, so we have that wavelength. In fact, what we did, we bought a photomultiplier with, you know, that the efficiency of a photomultiplier of the photodetectors is a sort of Gaussian, no? Okay? So that, depending on the technique, the materials of the photodetector, can be at some wavelength more efficient than others. So we chose the best photodetector having the highest possible efficiency in the wavelength we are using. Okay, so this is the system. Now, what happens in terms of first currents and voltages, after in terms of optical emission to our molecules? Of course, I'm using as an example this group of ruthenium, but can be extended to all the other electrochemiluminescent molecules. And I take the opportunity now to tell you another important thing that I forgot. The tissues, in general, have a fluorescence itself. So when, for example, I label a tissue, I want to do a fluorescent study using fluorescence of this tissue because inside I insert my dyes, my labels. I have a self-emission of the tissue itself in terms of fluorescence. So I put the laser and I have back the light from my dyes, but also from the tissue. So is again, for me, is noise, is background noise, because I have light from the tissue, so from everywhere. The other interesting point of electrochemiluminescence is the fact that very, very few molecules have this property. So, the tissue is not electrochemiluminescent. And so, again, I increase or I lower down the background noise, talking about optical noise, that I have from my system. So this is another point that is interesting talking about this technique. Okay, but here we have the voltage we apply, and here we have the current we measure. It is an electrochemical cell, so we can see it as a standard electrochemical cell where I apply, for example, here the example is from -2 volts to 2 volts, and I measure the current I have back, and the current I have back usually in an electrochemical cell as a hysteresis and as this shape, okay? The interesting point is that there are some points of my trans characteristics, let's say voltage on current on voltage, or my electrochemical reactions, in which I have the emission. In this case, the molecules showed are also other emissions in terms of light, but we are talking about this. So, the emission of the orange light, because what I'm doing is to stimulate using the counter, so I induce using the counter electrode my voltage inside the cell, and I do it what is named the cyclic voltammetry. So I start from -2, +2, I go back and forth, -2, +2, back, -2, +2. So I do up and down this, and I have back this response in terms of current, but the important point that every time I pass in front of this moment, when I have, yes, the current, because I have the reaction, because it is a redox, so it's, in this case we saw it's an oxidation, so I have exchange of electrons, and so I have the current. But in the same time, I have emission of light. So, the point is that in our system, every time I put a voltage of -0.8 volts, I have an emission of light. So, it's a periodic, remember this because it's one of the fundamental points we will exploit later. I have a periodic signal that is going back and forth, and every time I pass in front of -0.8 volts, I have an emission of light. My detector, my photodetector, has a light, and so it gives me back the signal that he received the light. Clear? Okay, so this is the concept, and let's also talk about, I told you, I'm not only focused on the sensor itself, I'm interested to build a system. So talking about the system, my specifications are, I want to build parallel systems, so multiple electrodes, not only one. You know, in these techniques, they use the biochemists, the biologists, they use an array of electrodes. Sometimes 96,000 electrodes in some cases, okay, also more. And must be a simple utilization. At the end of the product must be a one-shot use of mine, because in this case you are using blood, you are using you're doing clinical tests of these, using these sensors, and so usually they trash the sensor every time they use it, of course. For us, talking about research, we cannot do it because it becomes too expensive. So we are talking research, so I will reuse my samples, and so I have to deal between the possibility to reuse it, also if the final device will be disposable, will be thrown away after the detection. And so now it's also interesting to focus to reach the detection limit, to understand which are the minimum number of which is the minimum number of molecules that I'm able to detect. But can be an interesting information, but I write you here. Maybe we are, as researchers, interested in having the detection limit as much as possible lower. But maybe in some applications it's not needed, because I have the signal. I don't need to go too much down. So maybe I can save complexity, I can save money when I will build the application. Of course, now, as researchers, one of my goals is to measure how much I can stress my sensor, which are the limits of my sensor. This is, of course, an important information. And must be portable. So, we are talking, and now I mention now another thing in respect to fluorescence. Fluorescent laser filters is not a portable system. I cannot build a portable system. In this case, you see, electrode, photodetectors, and so on, I can think to build, without any problems, a portable system. How I label my DNA? I don't know if you are familiar, but I will just tell you a few words about it. I label my molecule. In this case, we are talking of a DNA, in this example. What I do, I put on the surface of my electrode what is named the probe. You know that DNA has a complementarity between the bases. So, thymine, the T, can link, can bond only with the A. I mean, the guanine, the G, can link only with the C, the cyanine. So, in this case, I can use this on the surface of my, I do it here so you can read it. On the surface, which is my electrode, I link with silane, with some chemicals, techniques, single strand DNA, so not, you know, the DNA is a double strand, okay? Where I put, for example, A, E, G, okay? Or longer, of course. For example, I give you only 3, but I put 11, 20, or so longer chains. And I put in my solution all these reactions happen on the surface of the working electrode. They happen also on the other, on the counter electrode, but with the less efficiency because of the system. So, but we are interested in detecting what happens on the surface of the working electrode. On the working electrode, we can use different materials, and this is the key that I told you when I started. We started using metal, and I will show you examples using metal electrodes, and then, this is the final solution I want to show you, we passed at the end to use also carbon nanotubes or carbon-based in general electrode as working electrodes. The rest of the systems remain almost the same. Or we chose to leave it as it is because of doing comparison. With changing only the working, all the system, the rest of the system stays the same, remains the same, and so I get the possibilities also of comparing the different solutions. So, talking about the electrochemical cell, I need a potentiometer, so the electronics for managing my electrochemical cells. And from the solution, we are interested in detecting the emission light, the light emitted by the working electrode. And here, now we are using a photomultiplier for detecting the emitted light. All of it is managed by the computer, a workstation. Okay? Please.
You detect only the intensity of the emitted light or also the wavelength?
No, only the intensity. Also because in this case, your question is right because it's related to the previous question. Up to now, we are working only with this label. So we have only one wavelength. You're right. And also in ElectroChemiLuminescence, the biochemists develop different labels emitting different lights. In that case, you need to also have a detection in terms of wavelength. But now, up to now, we did not start this, and we are focused on this molecule, this group of ruthenium.
But in fact, the wavelengths, the emission wavelength is defined by the molecules.
Yes.
So
this molecule emits.
You just have to take the intensity because you already know the wavelength.
Absolutely. Okay. Absolutely.
But in some years, you will be able to detect also the wavelength.
In case, but in that case, the solution can be to insert filters.
Okay.
Optical filters, because it's a unique point. So we come back to the need of optical filters. You spend more, but it is. And the wavelength of emission is strictly related to the molecule you are using. So we are using that molecule, so we have that wavelength. In fact, what we did, we bought a photomultiplier with, you know, that the efficiency of the photomultiplier of the photodetectors is a sort of Gaussian, no? Okay? So that, depending on the technique, the materials of the photodetector, can be at some wavelength more efficient than others. So we chose the best photodetector having the highest possible efficiency in the wavelength we are using. Okay, so this is the system. Now, what happens in terms of first currents and voltages, after in terms of optical emission to our molecules? Of course, I'm using as an example this group of ruthenium, but can be extended to all the other electrochemiluminescent molecules. And I take the opportunity now to tell you another important thing that I forgot. The tissues, in general, have a fluorescence itself. So when, for example, I label a tissue, I want to do a fluorescent study using fluorescence of this tissue because inside I insert my dyes, my labels. I have a self-emission of the tissue itself in terms of fluorescence. So I put the laser and I have back the light from my dyes, but also from the tissue. So is again, for me, is noise, is background noise, because I have light from the tissue, so from everywhere. The other interesting point of electrochemiluminescence is the fact that very, very few molecules have this property. So the tissue is not electrochemiluminescent. And so, again, I increase or I lower down the background noise, talking about optical noise, that I have from my system. So this is another point that is interesting talking about this technique. Okay, but here we have the voltage we apply, and here we have the current we measure. It is an electrochemical cell, so we can see it as a standard electrochemical cell where I apply, for example, here the example is from -2 volts to 2 volts, and I measure the current I have back, and the current I have back usually in an electrochemical cell as a hysteresis and as this shape, okay? The interesting point is that there are some points of my trans characteristics, let's say voltage on current on voltage, or my electrochemical reactions in which I have the emission. In this case, the molecule showed that also other emissions in terms of light, but we are talking about this. So, the emission of the orange light, because what I'm doing is to stimulate using the counter, so I induce using the counter electrode my voltage inside the cell, and I do it what is named the cyclic voltammetry. So I start from -2, +2, I go back and forth, -2, +2, back, -2, +2. So I do up and down this, and I have back this response in terms of current, but the important point that every time I pass in front of this moment, when I have, yes, the current, because I have the reaction, because it is a redox, so in this case we saw it's an oxidation, so I have exchange of electrons, and so I have the current. But in the same time, I have emission of light. So, the point is that in our system, every time I put a voltage of -0.8 volts, I have an emission of light. So, it's a periodic, remember this because it's one of the fundamental points we will exploit later. I have a periodic signal that is going back and forth, and every time I pass in front of -0.8 volts, I have an emission of light. My detector, my photodetector, has a light, and so it gives me back the signal that he received the light. Clear? Okay, so this is the concept, and let's also talk about, I told you, I'm not only interested focused on the sensor itself. I'm interested to build a system. So talking about the system, my specifications are, I want to build parallel systems, so multiple electrodes, not only one. You know, in these techniques, they use the biochemists, the biologists, they use an array of electrodes. Sometimes 96,000 electrodes in some cases, okay, also more. And must be a simple utilization. At the end of the product must be a one-shot use of mine, because in this case you are using blood, you are using you're doing clinical tests of these using these sensors, and so usually they trash the sensor every time they use it, of course. For us, talking about research, we cannot do it because it becomes too expensive. So we are talking research, so I will reuse my samples, and so I have to deal between the possibility to reuse it, also if the final device will be disposable, will be thrown away after the detection. And so now it's also interesting to focus, to reach the detection limit, to understand which are the minimum number of which is the minimum number of molecules that I'm able to detect. But can be an interesting information, but I write you here. Maybe we are, as researchers, interested in having the detection limit as much as possible lower. But maybe in some applications it's not needed, because I have the signal. I don't need to go too much down. So maybe I can save complexity, I can save money when I will build the application. Of course, now, as researchers, one of my goals is to measure how much I can stress my sensor, which are the limits of my sensor. This is, of course, an important information. And must be portable. So, we are talking, and now I mention now another thing in respect to fluorescence. Fluorescent laser filters is not a portable system not build a portable system. In this case, you see, electrode, photodetectors, and so on, I can think to build, without any problems, a portable system. How I label my DNA? I don't know if you are familiar, but I will just tell you a few words about it. I label my molecule. In this case, we are talking of a DNA, in this example. What I do, I put on the surface of my electrode what is named the probe. You know that DNA has a complementarity between the bases. So, thymine, the T, can link, can bond only with the A, amine. The guanine, the G, can link only with the C, the cyanine. So, in this case, I can use this on the surface of my, I do it here so you can read it. On the surface, which is my electrode, I link with silane, with some chemicals, techniques, a single strand DNA, so not, you know, the DNA is a double strand, okay? Where I put, for example, A, E, G, okay? Or longer, of course. For example, I give you only 3, but I put 11, 20, or so longer chains. And I put in my solution all around the DNA that I want to detect, okay? And this DNA is labeled. What this means? It means that in some place, of course, usually at the end of this strand, I have the, you can see it here, the ruthenium molecule. Okay? So, I put here the label, and I have from the other side my DNA, the sample of the DNA of the patient I want to analyze. And it's not the case, of course. This DNA that I want to detect can be C, for example, A, T, and the other is longer, of course, but I have the complement of what I put on the surface. And so I put here a perfect hook to catch and take my molecule. So my molecules, because they have the complement here, they link, because here I have my C, here I have my A, and here I have my T. And they are labeled. So, one of the mutations that can induce diseases, genetic diseases inside the people, it can be, one of the examples are also more complex, of course, mutations, but I give you the simplest example just to understand what we are talking about. It is a deletion. I do not have, I'm sick of this, I have this disease, I do not have this part in my DNA. Means I'm maybe not sick, I'm simply a carrier. So for women it's important to understand if you are carriers, or also for men, because after you have a mix of the two genomes. And so it's interesting to understand if I do not show up that I have this problem inside my DNA. But I can understand it because I do this test, I understand that I do not have this part. Because if I have it, the molecule links. After what I do after the linkage, I clean, I put out all what was not attached, because these bonds are efficient, are good, so I can clean, I can wash out all the DNA that is swimming around my sample, and now I hope it's clear, if I see a light, it means that I have the bonding, and so means I have this piece of DNA. If in my DNA I do not have this sequence, I do not have any molecule attached on the surface of my electrode, and so I do not have light. And this gives me the information I need. This test must be also not only qualitative. I told you qualitative. Yes, no. I have it, I do not have it. But if I work with arrays, with large amount of electrodes, there are also some techniques for having this information. You know, if I make a carrier of something, means that I have one chromosome good and the other chromosome not good. So I have the half of the quantity of DNA of a normal person. So the quantity of the emission, if I have a very shining light, okay. If I have, it's not half, you know, in reality it doesn't exist, this difference, 1/2, but okay, around 1/2, but at least a difference in emission of light, I understand that I'm not normal, I am a carrier of this disease. Yes, I feel. If I do not have emission, means that I'm not only a carrier, but I do not have this piece of DNA. Please.
So in case of mutation, for example,
Yes.
there's some elements which is missing. So you said that A connects to the G
Yes.
and the T to the C.
No, T to A and G to C. Yes, but doesn't matter. Okay.
Doesn't matter. So there's something missing, or it could be also some kind of crossing. So you see what I mean?
Yes. In this case, it's again interesting. This maybe because the example is correct. I have a mutation. So my strand of DNA has, again, I'm talking about a very short, but we must talk, of course, of longer sequences. But I have the T, but for some reason I have T, G, TGG. This was, well, what I expected was T, A, C, no? For the complement. But for some reason, yes, a mutation, these two bases are two Gs. It's an error. So what happens? This strand, always labeled, can link here using the T. Okay? And here I have the label. But the energy of a link is very much lower than when I have all the link. Okay? So, and here again, biochemicals, biologists are good in it. They can give enough energy when they wash out the things to detach the strands that are not attached so well. And this is the simplest. There are also more complex techniques. I can go in details, also because I'm not the expert of it. But just to give you the idea, okay? So, I have a possibility because the bond is not so good. When I wash out. But, and here I arrive what I mentioned, 1/2. I have always mismatches. I have always bad links, because you can imagine, here I have bases, so also other strands that do not have all the sequence can attach. And sometimes maybe they can attach and do not be removed when I wash. But again, if I have a good statistic, if I have an array of a lot of electrodes, and I wash, statistically I can understand if I had a good bond, so I have without problems my DNA, or depending on the emission, understand if the signal, the light I have is just due to mistakes, just due to bad bondings, bad molecules attached on the probe, or are due to real problems related to mutations. Did I answer?
Yeah.
Yes, please.
How is the labeling made? Is every sequence which is flowing around, which has a T at the end, is automatically labeled by the labeling process? Or?
Usually, one of the techniques, there are again several, but the one I know better, it is based on the PCR, so the polymerase chain reaction. That when I duplicate the DNA, I have my strand here, E, A, C, G, and so on, and the polymerase is the enzyme who does the work, because all around I have A, I have G, I have single bases that are flowing, okay? The polymerase take, for example, the A and attaches, I do it very simple, well, biochemist maybe, of course I can go more in details, but again, the concept, okay? I have the adenine here, after it takes a T and put it, so. What I do is to put in solution on also some, for example, T, who are labeled. So, when I insert a T, I have a label attached. And what they also do is that when I attach this, the enzyme is blocked, cannot continue. So I will have a lot of sequences ending with T, and maybe this is here the one what I want, but I have also my strand and PCR, the reaction, the polymerase chain reaction. It is also designed to duplicate, to replicate this strand, this part of DNA. So I will have in my solution a lot of strand with the sequence I want to catch labeled. Or there are also single S and E, if I remember well, single nucleotide extension. So I extend what I linked here with a dye with a base that is labeled. So there are, I tell you, several possibilities. But in any case, very important that I can reach with these biochemical solutions, I have the pieces I want to catch correctly labeled. Just, okay, in general. Good? So, coming back to our point. This step means that I have a mix of chemistry, biochemistry, and technology that I must put together. And, of course, me, I'm here. I want so realize a microsystem. I want to realize the hardware necessary for interfacing my microsystem, my electrodes, my emitted signal I have. And I need to set up a workstation for managing the experiment, and I need to set up 2 different levels of software. So firmware for the control of the boards, and also standard process software on the PC. And this is in general the system I have. The microsystem, the microelectrodes, the interface system, and the analysis and management workstation. What we did, we started from, I tell you now, I start with methods, okay? So, we built different shapes, rounded, squared, we also built stars, okay? for testing the geometry, because the geometry of the electrode is also important. And if you know, in terms of current, in the corners, in the shape that are stiff shapes, I have always an increase, the density of the current is higher. So, and you see that this emission is optical, but is related, of course, to the exchange of electrons, so to the current. So also having maybe square, having angles, having this tried star, okay, for detecting if the emission, the efficiency of emission was better. It was just, we did this type of test. And in this case, this work was done in the shape of one Italian, was funded by Italian government, named Lateral. This Lateral project, with which one of the partners was the Olivetti inkjet. So they built these different type of electrodes, and we used, you see here, this is the reference electrode. So I told you 3 electrodes, but here you see only 2, the working and the counter around. So in this case, we did not integrate immediately the reference electrode, we put the reference electrode outside, close. It was not important for us at this stage. And I want to show you what happened in terms of emission. And so you see here the electrodes, you see it? Yes. So now we close the chamber, so we are in the dark. We put just the geometry just to show you where is the electrode, because now we are in the dark. Now everything is closed. We apply the voltage, and you see you have the emission periodically. Now we are passing in front of this voltage. Now we are moving up and go back and forth the voltage, and every time we passes in front of this minus 0.8 volt, we have the emission of the light from, you see, from the center, so from what is the working electrode. Now, okay, we open. I want to also show you, we did also another test with the square. So we have this shape, this square, and see here again, we close the chamber, we apply the voltage, and we have the emission. In this case, the emission is lower because of the condition of the test, but you can also see one thing here, that every time I pass back in front of the emission, the quantity of light is every time, every cycle, let's start to call them cycle, every cycle it is a little bit lower. And this is one of the keys that I will tell you later. Okay, so this is what happened in terms of emission. Let's pass, and after we can do a small break. I don't want to give you too many informations in one time. Let's talk about the system. The system we produced, in this case, Olivetti inkjet produced an array of 16 electrodes. We built the electronics for the hardware interface with this channel management, and also the software for elaborating the system. So here, before we just had the names, now you have the devices. So we have we had in this case our electrodes here with the contacts connected to the interface board, and then to, in this case, a laptop for controlling the experiment. And the board at that stage, at that time, now we did improvements, and I will tell you later, but we had 2 inputs of 8 channels each, so we managed all the 16 channels for controlling. So here we have the different channels of the relay for controlling the channels. We control the electrochemical cell, and on the top we use a photomultiplier, and then we have the workstation. And this first release of the software was done in LabVIEW. Now, the software we are using now is based on Python. We changed it to Python language, but of course the concept is the same. We have possibilities to choose which channel you want to use. We have also the possibility to choose the photo detector, because we characterize the photo detector because we have a direct comparison between the voltage I have with photomultiplier. I have a voltage of measurement. The voltage is related to the quantity of light I have. So depending on the specific sensor I'm using, I know the quantity of light because we characterize it and we calibrated our system. And then the waveform I want to apply. I just gave you, because it's the unique example we need from an electrochemical cell, so the triangular stimulus, because I go up and down, and we use from -2 to 2 volts, okay? But in electrochemistry you can also use more complex stimulus waves, stimulus voltages, ramps, square waves, but in our case is not is not needed. Okay, we simply need, but we also decided to do in the software a possibility to apply different other also typologies of waves. And here you set up how much from the start, the end, how many cycles, because we can also, and it is one of the things that I will go in detail soon, also we decide how many cycles we want to have after ending, before ending our experiment. And here are the results from the system. So we have the AV, so the current of the voltage. We have the measured voltage from the photomultiplier related to the applied voltage. We have the emission in light in time. This is the time. This is the voltage I read from the photomultiplier, so the emitted light. So with all this graph, all this information, we can understand how is going our experiment and how is working our experiment. You see here, okay, the IV characteristic, and I take this to tell you what I mentioned before. Now I hope it is clearer. I told you, we use not a standard reference, the silver chloride. So the emission of the light, it is at a precise voltage, but if I change the reference electrode, it is almost 100% possible that it changes. But for me, it doesn't matter. My goal is to have a light. If I have it at -0.8 or -1 or 1 volt plus or plus half a volt, for me, in my system, in my experimental setup, I have emission there. It is my information. After that, I know it. I know that I have emission there, it's enough. So I can simplify my system having the same information I would have in a more complex system. So this is why, now I think it's clearer, why we did not choose the silver chloride, because yes, I have always the voltammetric response, I have always my electrochemical cell working, simply maybe is shifted a little bit, but for me is not an important information. Okay, so now we can have a small break, because after we start to enter in the more specific topic in which today we have to talk, so the use of carbon nanotubes and carbon-based materials. Okay?
Great, thank you.
Welcome.
So maybe 20 minutes break, it's sufficient to take coffee and a cafeteria. Come back. This is good, 20 minutes.
For me, you are the boss.
If you need more, we can do more.
No, it's okay. What we call carbon. It is because the reason was one of my colleagues in Politecnico, is able to produce carbon nanotubes and carbon-based materials, but mostly carbon nanotubes. In fact, he's the leader of the carbon group of Politecnico di Torino. And we discussed a little bit for saying that around we were talking about a few years ago, there are works in which they are using carbon nanotubes as electrodes for electrochemical cells in general. And so we decided to try carbon nanotubes inside our cells, and our ACL cells. And we discovered that the emission of these carbon nanotubes is very interesting. And I don't tell you now because I want to show you some numbers. The 2 points were these: efficiency. The efficiency of the electrode, we have seen that is very high, very good, because carbon nanotubes have a very large electrode surface, because they are not as a metal flat and continuous, but you have this forest. In this case, you will see we produce forests of carbon nanotubes, and the total surface of the electrode is dramatically increased. And from the other part, I told you about these cycles. The interesting point was that the emission was constant for a long time. So we did not measure, and I will show you results, the loss in efficiency we had measured before with metal electrodes. And so we decided to use them. The experiment in which we are not yet at the end, but I will show you what we did in terms of carbon nanotubes, is this one. Is to use our carbon nanotube to put on the top of the carbon nanotube a fluorescent. You told us a lot of things that ACL is better than fluorescence. Yes, the fluorescing for us is just first a linker, because the biochemical people who were working with me, very expert in put fluorescing on the top of carbon nanotubes, so we decided to use this, because on the fluorescing they are able to build a cross-linker, and we exploited 2 possibilities, one with hydroxyl group, the other one with amino group, for doing the link to the DNA probe, what I previously showed you. So my DNA probe, and then on the top the lutetium that is variable. Okay, this is the final structure of the experiment we are developing. Let's start from the electrodes. I show you the result, and after I go back. This, my colleague of mine, the name is Tagliaferro, as I told you, of the carbon group of Politecnico, they are able to produce cylinders of bundles of carbon nanotubes. So we have a lot of carbon nanotubes there, and they produce them starting from silicon wafer. They spin a photoresist for doing a photomask, and so for defining the areas, the zones where they will grow up the carbon nanotubes. Then, after the development of the photoresist, they deposit metal on the top of the wafer, they do the lift-off, and they are able to grow up the carbon nanotubes where we do not have the metal. So they grow up the carbon nanotubes directly on the surface of the silicon dioxide. Please.
What kind of technique is used to deposit CNT?
CVD. They use CVD. If you are interested, I have some papers where it is also explained how much they have now 2 CVD machines, one older, one newer, on which they control also all the environment of the processes, but the concept basically is CDD. And they start from the normal, now I'm losing what, they are starting from something very simple. The process is very simple. Now, after I have it here with me, I can show you. Camphor, from camphor, okay, I forgot. They use as a carbon, from which to extract carbon, they use the camphor, normal camphor that you can buy in the supermarket. So they had as goal to produce a very low cost process. And so they grow these carbon bundles of carbon nanotubes attached on the surface as in this picture. Okay? So they built arrays of these, but we don't use them as arrays because they do not have a metal contact, because after, of course, we put out the metal, or it's not in, but the metal is not useful for doing interconnection. So this cylinder must become an electrode, and to take this cylinder that we detach from the surface for building an electrode, we use this technique. We have, and this is done directly by my guys, we have our carbon nanotubes on the surface of the silicon. We put on the back some, I forgot. Now we are working with electrodes of a dimension of 1 millimeter or half a millimeter. Most of the experiments I will show you are based on the diameter of the cylinder of 1 millimeter. Carbon, the bundle, yes. Yeah. The diameter of the bundle is, it is now, most of the experiments we are doing that in 1 millimeter. Of course, the goal, the final goal of everything is to arrive to a miniaturizer, to a real microsystem. Here we are not talking of a real microsystem, where what they are working, Tagliaferro's group, is to grow up the carbon nanotubes directly in metal, that is not trivial, and so to have immediately your wires on the top, the carbon nanotubes, in this case also with a smaller dimension, because now that I showed you here, we cannot manage two small electrodes because we do this by hands. But the final goal is to produce something in a micro dimension. But we put some silver paste on the back, so we connect a wire, a standard wire. Then we put PDMS all around with, well, not all around, but we create chambers inside the PDMS. PMMA, we use PMMA here as mask, we can say, and after we put the PDMS, okay, in the wells of the PMMA. We wait that everything becomes more solid, and after we can detach this structure that we produce for giving, at the end, yes, the connection, but also a mechanical structure to the electrode that we can use. As, of course, you can imagine a bundle of carbon nanotubes alone will be broken in a few seconds. So in this case, we give a mechanical strength to our electrode, and in the meantime, we create the wire. It is, again, not the final goal of the project, but was to start to did experiments, for starting to do the test we needed for having the idea if this system is effective, is working. Okay, so, yes, please.
Can you talk what is about the previous one?
Yeah.
These are carbon nanotubes, which mean
yes, you see,
inside it's.
You can. This is a carbon nanotube. You see, you have them. The the process is good because they are oriented. You don't have perfectly shapes, but they are oriented.
You are always talking to use them as electrodes.
Yes.
I never heard that we think it's interesting to use them like microfluidics or nanofluidics tubes.
In this
why,
case,
why do not take advantage of this property that they are tubes, which is empty inside?
okay.
Electrodes, we can have electrodes with different materials also. Maybe not with these dimensions.
Yes.
But,
Okay, 2 points. The first, I will show you very soon, in the next slide, why they are interesting as electrodes. Not talking about nanofluidics, just for using them also as large electrodes if you want, or to use them as standard electrodes. Because in this case, you exploit this possibility that they are empty, that they are, you see, not regular, so all your molecules can enter, can be the surface, as I told you, of the electrode is larger. So you exploit these, not for the fluidics, but for the surface of electrode that is large. Talking about nanofluidics and all this, there are some works in literature about this. I tell you, from this side, can be interesting, by sure, because if you are able to produce one carbon nanotube, having the liquid that is passive, it is a single carbon nanotube and electrode. But you are becoming from a company. Is it useful? So, I mean, maybe it's too small for some application. But I don't say that is not useful at all. I mean, has to be decided. From my point of view, I tell you, it's an interesting field, but in this case, it changes the application. In this application, I don't need to go so deeply in reducing the dimension. Because at the end, I must have an emission of light, I must have from not only one carbon nanotube that can be maybe too low. Okay, but again, your idea is good and it's possible. Yes. Yeah, for example. Yeah. No, it's absolutely a good idea to go ahead and to study. And there are already some studies in the field. But for my goal, this is the point, and so I answer to your first point. You see here the emission of same dimension, 1 millimeter in diameter of a gold electrode. You see that after 3 cycles that I go back and forth, the emission is going down rapidly. With the carbon nanotube, we had this result. So, means it is almost stable. Of course, as always, it's not absolutely perfect. It's an experimental result. But we did hundreds of cycles without, after a while, of course, after 300 maybe, or it depends again on the experimental setup, but after you start to lose in efficiency. But the efficiency is absolutely great in terms of emission. And so what we had in mind is to say this. It is better, of course, after a proper data treatment, but this improvement, because the reason of it, I'm not able to tell you a final result. I mean, we asked to some chemists, some experts in carbon nanotubes and electrochemistry. There is a professor that I know very well in Bologna, Francesco Paolucci. But we have some ideas of why it is happening. I tell you, the first time we thought that this was because of large surface electrode of the carbon nanotubes, and so you have always a renewed surface, because you do not have problems of a surface of electrode that maybe is polarizing or having some problems in terms of efficiency. But after, and this is the second step I will tell you today, we discover that with other carbon-based materials, we have the same behavior. And so means not carbon nanotubes, means carbon, this interesting behavior. And this so can be related to hydrophobicity of materials in terms of, can be due to other reasons, but to be frank, I did not find yet around where someone can take you, this is why. The guesses are more related to the hydrophobicity of the carbon in terms of the sample we have around, and so maybe this hydrophobicity helps in having the surface always fresh, clean, more clean. Okay. So we are arriving from an electric. Yes, please.
Yeah. So in this case, why with gold electrodes the signal decrease?
Because of the property of the material. So
it's a kind of oxidation.
By sure there are oxidation, there are polarization of electrons, there are, and in this case, electrochemists have a lot. They have a lot of information on metals, and it is normal. It is what you expect. This that you lose in efficiency. With carbon, you don't have this lose. You have it, but the time constant is longer, very much longer. Also because, but now after your question I must tell you something in advance of what I will tell you later. You have with metals a similar behavior when you work in flow cells, so in flowing liquids. So your liquid flows on the top of the electrode, so again it is maybe the electrode always more fresh. I'm talking, I'm not a chemist, so in terms of freshness, in terms of the electrode is efficient for a longer time. But I told you, I'm not able to give you a chemical specific and final solution. Okay? This is what we measure. And because we are arriving from electronics, from telecommunications, we said, okay, we can exploit the periodicity of the signal using averaging, locking algorithm. I don't know if you know them, but are specific algorithm filtering or averaging for extracting from the noise your periodic signal, because we have a periodic signal, because at each cycle I have the emission in that specific place in time, okay? And so if you do an averaging of 100 of cycles, the noise is random, so you have the noise spread all around. But if you do an averaging, and I will show you the result, you extract from the noise your signal in that precise time in your cycle. Because you know that if we use, if I remember, we use 17 seconds of cycle, in between you have at second number 7, I don't remember, which is, you have emission. So you know that that is the point where you have emission. If you do an averaging, mostly, after we also apply some filtering, some other, but the key is based on averaging. And you extract, you increase the signal to noise ratio. You extract the signal from the noise. And this is what I told you. This is the raw signal, so what we have from our photomultiplier. We apply the algorithm, so we have back an embedded system, we apply in real time measuring, and this is the result.
So it's a lock-in technique.
It's similar to a lock-in, but it's not a lock-in. If you know a lock-in, we tried also the lock-in, but the averaging at the end sounds better. But at the end, the concept is a lock-in. Also, no, it's a different algorithm, different technique, but you have, because of the periodic signal, you can apply also the lock-in. Okay? And so, in this case, you see, we have the emission now, we see about the 12 seconds inside the cycle of 16 seconds that we go back and forth, and you see, this is what we obtain. So we extracted from this signal that is completely noisy, a very good signal, very clear. And in terms of numbers, these are the results. We arrived using gold electrodes up to a dilution of 1 to 50 from our stock solution. Using carbon nanotubes, in this case, this is the result, we arrived up to 1 to 1000, so 40 times more. A very great increase. And in terms of concentration of ruthenium, I did not tell you. These experiments were done using simple ruthenium on the top of the electrode. We did not put DNA and everything. We simplified the system because I was interested in understanding the efficiency of the reaction, the efficiency of the electrodes. So, in this case, we arrived at 1 to 1000 means 80 picograms per microliters concentration. And this is one of the statistics. Now we are generating new statistics, but you see that I told you 1 to 1000, but this was the result. This test was done with the dilution of 1 to 1000. So you see, we have the possibility to go also further because it's very, very clear, the signal. In fact, doing the statistic, we arrived up to 1 to 2000 in terms of detection limit. And so now, talking about the biomedical application, what we did, and I will show you some results, and we are doing is this. I told you, we put the fluorescein on the top of the carbon nanotubes, and now we are again skipping the DNA, but we are attaching directly the ruthenium just to have the detection of the technique. The fluorescein, in this case, is also interesting because, yes, it is for us a linker, but it is a sort of reference because we use fluorescence for understanding if ruthenium is attached on the surface. So we use a standard technique for proving that, yes, you was able to attach the ruthenium on the surface of carbon nanotube. And after, we use ElectroChemiLuminescence for doing the experiment. And these are the results. Let's comment these results because it's one of the starting point for what I will do next. You see, we used amino group or hydroxyl group for doing the linking. And the efficiency was very different from the two. You see, using amino group the emission was very good. In terms of using the COH group, you see, was very much lower. And this is the measurements we had with 7 different electrodes. From these plots, I have the bad point of this technique. That the reproducibility is a mess. The reproducibility is very, very, very low. Here we are talking of an emission in an arbitrary unit because it's normalized, but 0.9, 0.2, so it's unreproducible.
Because your electrodes are all different.
Because of it. Look at the surface. The surface is a mix of carbon nanotubes, But on a 1 millimeter dimension, of course, the carbon nanotubes present on the surface, I cannot count the number. So, the idea is good, because I have a very high efficiency. I have this type of interesting emission, periodic emission, but with these dimensions, the reproducibility is impossible to have it. So the solutions are many. The first one, of course, is to reduce the dimension, and it is one of the goals. So the carbon nanotubes, if I go to integrate the carbon nanotubes, put it directly on the top of the metal, I can go to have maybe 10 microns, 50 microns diameters for my bundle. Okay? So this, by sure, will help. I don't have the results now because my colleagues are working for preparing them. But in any case, also in 500 microns, you have a lot of carbon nanotubes. So again, the reproducibility is not absolutely perfect. We can go in the direction that he mentioned, so to work with very few, when you want few nanotubes, but in that case, your technology, and I tell you, up to now, I'm not able to reach this point, okay? So, all these considerations, also because we tried, and we wanted to try other type of electrodes, gave us the idea to say, okay, but if the carbon is the key, let's test other carbon-based materials and see what happens. And so we did, and we are working now on glassy carbon, silicon carbide, and not yet, but if I will find someone able to give me good graphene electrodes, I'm here ready to react and to use them. And we obtained this. We bought from Japanese companies, so in this case is a standard and commercial glassy carbon electrode. And we had this emission that this can be also sometimes happens also with carbon nanotubes. So you have a good efficiency at the start, a losing efficiency, but after they stays for a long time. So again, this periodicity we have, okay? Also with glassy carbon. But look at it. This is the efficiency in terms of dilution of, sorry, it's yellowish, you don't read it very well, of a carbon nanotube. And this is the emission, again, in millivolt on millimeter square of a glassy carbon. The efficiency of emission of the glassy carbon is absolutely nothing in comparison to the one of carbon nanotubes. I did not put also the plot of it. I can tell you that it is absolutely reproducible. You obtain almost always every experiment, every time, same numbers. So you have an absolute reproducibility of it. But efficiency is dramatically lower. We tried also, this is a cooperation we have with the University of South Florida in Tampa, with Professor Steven Sandow, and he is working on silicon carbide for other applications. And he sent us some sample of silicon carbide, and we did also experiment, and they are more noisy from this side, but again, we obtain the periodic signal we want. It means, okay, it works. From the other side, and they gave us different type of polysilicon carbide, and monosilicon carbide on silicon, and monosilicon carbide on SOI, so different possibilities. And we have seen that there are, you see, the efficiency of a monosilicon on silicon on an isolator is better than the other, so we did also some tests, are preliminary tests. This is why I do not go into much details, because we are now in the middle of doing these experiments, and but we also check which were the best solutions, the best performances of our different possibilities. And we went in the direction of silicon carbide because silicon carbide seems one of the most biocompatible materials in the world, in now, let's say, on the market. So, these are biomedical applications, so of course to have a very good biocompatible material is you can imagine a great possibility and result. So, also silicon carbide works. In the meantime, and I already spent a few words about it, we are also doing some flow injection analysis test. So, inserting our electrochemical cell, and in this case we started again with metal, because we started with standard solutions and then we moved to the more specific solutions. So we produce an electrochemical cell inside a PDMS microfluidic device, and we did some measures, and in this case you see that the static measurement in comparison with the fluidic measurement is better. These results are not so good. Again, we are in the middle, these are preliminary results again, but we are starting to have the proofs of what we expect, and also the static versus the dynamic measurements. You see here the signal to noise ratio in 0, this is the flow, the speed of the flow, 0, so this is the static, and here increasing the flow, we increase the signal to noise ratio. Also because, as I told you, in this case we have, but you can see it here, again, it's not very good, we must improve the electrochemical cell and also the fluidics, but is again more constant and higher. And here you see you are losing from this side the emission of the signal. So it proves what we expect. And so you see here the increase in the signal to noise ratio because of the fluidics of the measurement in flow. So this is what we did. Just a few final remarks about this work. Carbon-based materials are proven from the test we did to be very good candidates, okay, for ECL detection system. But if you are not interested specifically in the ECL is electrochemistry. So I can extend this property also to general electrochemical, okay, measurements. Of course, we must compare the different possibilities, and it is the work we are doing now. We are looking for different carbon-based material, as I showed you, and to test them. All these have the goal to integrate, to arrive at the end to a microchip, to a microsystem. Okay, I do not forget this. And I already told you that silicon carbide is one of the best biocompatible materials now. And also, for example, now we received a few weeks ago from a company in Japan some carbon nanowalls. So now the study on the carbon, you are, in this case, maybe also more expert than me in terms of materials. The study of the carbon is a very large area of research, very large topic, and people are trying also to exploit different shapes. And in this case, one of the promising shapes and geometries are these carbon nanowalls. So walls of carbon materials, of carbon-based materials, for again producing electrodes and this type of applications. So of course, all what I told you, I always say, I'm a professor, I talk. My guys, they are the ones who work really on the things. So I must thank them because of the, well, I hope the good results we had. So these are just few of the people of my group who are working. And in any case, I thank you, but I have in Torino a backup solution in case my ECL solution doesn't work. Because Lavazza is a company in Torino, and they are producing the ECL machine, the espresso cappuccino machine. So in case if I will not be able to use ECL in biomedical application, I can ask Lavazza if I can be useful for, because this was what I found going around the internet for looking ECL cells, ECL machines, and I found the espresso cappuccino Lavazza machine. So, and it is in Torino by case. And so can be a backup solution for our work. And of course, thanks a lot for your patience. And if you have questions, I really thank you because you also gave very interesting questions that I'm here to continue the discussion. Thank you so much.
Thank you.
So, other questions? Other things?
Yes.
Yes, please.
When you do the measurement, you detect the intensity.
Yes.
But do you integrate over the signal, because the intensity, you say it comes at minus 0.8
Yes.
volts. So do you only take the maximum peak, or do you integrate the signal which you get around 0.8 volts? Because for your carbon nanotube electrodes, I think you may have differences in the work function.
Yes.
And when the work function changes, you may have a signal which is broader in some cases and on a smaller voltage area in other cases. So
Yes.
you should maybe take the integrated signal.
Yes, you're right. In fact, this is what I have back.
Yeah.
Okay. Also because, as I told you, our setup is not perfect in terms of electrochemistry. So our cell, from an electrochemical point of view, it moves a little bit. Okay, it's not totally stable in terms of voltages and so on. So you're right. Now, in the first, you can do two approaches. You have this back from the algorithm. You have, of course, a peak where you have the highest probability to have emission. But, you're right, just take the peak is not the best. You can go further more deeply in extracting the signal. Because, you're right, around here, you see, it is not a very stiff and small and very not large peak. It is a little bit broader because you have a broadening of emission around this point. So, to also do some integration to measure the area of this, I'm not doing it now because I'm doing, you see, comparisons, so I'm not going to now deeply again do the last increase in the result. But absolutely, yes.
I mean, it could be a reason why you have so big differences between different carbon electrodes. Because they are not homogeneous and you have maybe areas which have other work function than others, so I don't know.
No.
No, because it's electric constant.
No, the difference between the carbon nanotubes is related also to the fact that during the experiment after we found pieces of carbon nanotubes around. So I did not tell because maybe of course you can improve it, you can increase it, but you have seen the surfaces are very different. So you can say, yes, I can do a sort of calibration, no? The dynamic calibration. But at this stage, with these dimensions, I tell you, you cannot do anything, because it changes every time too much. So if you want to work on carbon nanotubes, you must do two things. The first one, because now Tagliaferro is increasing also the quality of as much as possible unique or with a better geometrical disposition of our carbon nanotubes, more regular. So you must work on having a better regularity of your forest. Second, you must decrease the dimension. So if you lower down the dimension, for sure you will reach better results. And the proof of what I'm telling you is that with glassy carbon, absolutely reproducible. With the glassy carbon, I have a lower signal, but it is reproducible, 100%. Okay? Good.
You said when you talk about graphene, how come
Yeah.
the evidence from experts with this
Well, graphene is, I think, a good deal between good reproducibility, because if I do a layer of graphene, it is absolutely, well, I have not a perfect geometry, because I do not have a layer of graphene perfect. I have domains, I have, of course, but I can say that for reproducibility, I expect that it is better. From the other side, of the intensity of the emission, I expect it in the middle between glassy carbon and carbon nanotube. Because we know, for example, I don't know if you worked on supercapacitors, but at the end, the graphene has a larger electrode surface in comparison with other materials. So again, we exploit the geometry of the graphene because gives us back a higher surface, talking about the same dimension of the electrode. So you increase the efficiency, by sure, of the electrode, but from the other side, you have back also a better reproducibility. So this is why I see graphene also.
There's nobody doing this manufacturing.
Up to now, I did not find applications in ECL. Okay, I did not find yet, but I'm sure that some people are working on it. Maybe simply I have to renew my state of the art, because I'm sure because it's very interesting. So this is why I'm looking for someone who can which cooperate for doing this type of experiments. Okay? Good.
So thanks again.
Thanks to you. Have a nice exam now.
How much fresh air do you need?